Yeah, so thank you very much, Alexandra, for the introduction. And I would like to thank the organizers for inviting me to give this talk. It's a pleasure to be back in Paris for me. And also, all the more that I was actually a student in one of the previous editions. I think I should remove this. Yes, okay, sorry. I was a student in one of the previous editions, so it's even nicer. And so my lab works on evolution at the scale of microbial populations and on evolution at the scale of protein sequences. Today I'm going to tell you about work at the scale of protein sequences. And so my lab is at EPFL in Switzerland. And the work I'm going to tell you about today is on how we can extract information from protein sequences about evolutionary constraints, structure, and interactions. I will mention structure a little bit because it's at the basis of many of the methods, but personally I've worked rather on the inference of interactions and on also the inference of functional constraints. So before I tell you about this, I would like to introduce the topic of proteins. And proteins can be described at different levels. So let me see if, yes, okay. So the first level of description that we can take is the level of sequences. Proteins are heteropolymers of amino acids. And so if you know which amino acids you have along the chain, in principle, you know everything that there is to know about a protein. And the sequence can be represented as these letters. Each letter represents an amino acid. There are 20 natural amino acids, so 20 different possible letters. A protein that has a given sequence folds into a specific 3D structure, which you can see here in two different representations. And a protein also has a specific function, which is its biological role in the cell. For instance, a protein may be an enzyme, a molecular motor, or a receptor. And something very important in its function is also with which other proteins and other molecules this protein interacts. So the sequence fully determines the structure and the function. However, there are other arrows that I've shown that, from function to sequence and from structure to sequence. And these arrows come from the fact that the set of sequences that we observe in nature is deeply constrained by structure and function. What I mean is that during evolution, mutations may affect the sequence and change an amino acid. However, natural selection will be at the level of function, because function is really what matters for the cell. You want to have a good enzyme, etc. So selection is occurring at this level. And because of this, we find important footprints of function and structure on sequences. So sequences can tell us about constraints from the need to fold and the need to function. Something that I also want to emphasize is that I told you that biological proteins fold into a well-defined 3D structure. In fact, this is something that is quite remarkable. If you consider a random, if you consider a random protein with a random sequence where you have chosen, these amino acids here randomly, then it will not fold properly. This has been shown experimentally. And from a theoretical point of view, you can realize this also by seeing that these interactions between the random amino acids are essentially random. And this means that it's a complex system which is likely to have glass-like behavior and many locally stable low-energy states. So the fact that proteins fold into a well-defined 3D structure is remarkable, and it's a special property of natural proteins. Okay. So a very nice thing about this field is that we have a lot of data. So here I'm showing you a recent plot giving the number of sequences with time, which is coming from one of the databases of nucleotides. So protein sequences are usually obtained by translating the nucleotide sequences from genome sequencing projects. You can see here that the y-axis is logarithmic, which means that this almost linear growth means that, in fact, the number of sequences is growing exponentially in time. And currently, we have more than 10 to the 9 sequences. So this abundance of data is a great opportunity for methods which are based on statistical physics, information theory, or machine learning to learn about proteins from sequences. I will show you also another plot which is slightly older, but which has the advantage of also showing manually annotated sequences in green, and structures known experimentally from, for instance, X-ray crystallography in red. And so what you can see on this plot is that most of the data we have about sequences is just sequences, right? And all we know about them is coming from bioinformatics, and automated annotation, and our analysis of sequence data. Okay, so a lot to learn about this data. And so far I told you that there are, okay, more than 10 to the 9 sequences, which is huge. However, this set of sequences is organized into families. And so a protein family is a set of homologous proteins which have similar structure and function, and which have in fact evolved from a common ancestor. So for instance here, you have this schematic of a simplified alignment. All these proteins have sequences that are somehow similar but not exactly the same. You may imagine, for instance, that the first one is a human protein sequence, and the second one is its homologue carrying out the same function in the chimpanzee. Because we have a lot of similarities in our genomes, these sequences are similar, but not exactly the same. So that's the idea of a protein family. By looking at the statistics of this alignment, we can learn a lot about this protein family. So this is just, a schematic example. Here I'm showing you this time an extract from a real protein sequence alignment. So here, okay, I should introduce maybe some notations. So I'm going to write these alignments as matrices, and the number of columns is going to be called L. So that's the length of the protein, the number of amino acids, right, in the chain. And the number of rows is going to be called M. That's the number of sequences that there are in this family. So here, in this extract that I'm showing you, L is actually complete. This is a domain from a protein, so a subpart. And within this domain, I'm showing you the whole length. However, the number M is here very small. You have to imagine that in practice, we would have, let's say, 20,000 or something of these proteins. So that's an extract. And we can see that these sequences have some columns where you have a lot of similarity. So here I highlighted one of these columns. You see that there is always H, which is the amino acid histidine at this column. This is called conservation. It means that this amino acid is probably very important for the function of the protein, that we really need to have a histidine here. Okay, so that's a one-body statistic, and it's something that we are going to use. So let me just write this. One-body statistic. So we are going to focus on one column, for instance, the one I highlighted in blue on my slide. And this column is going to be called I. So I is between, okay, I should write it here. I is between 1 and L. And at each column, I mean each sequence of this alignment, we can have a state, which is the amino acid, and I'm going to refer to it as alpha I. It's the amino acid that is in I in this sequence. So alpha I is going to be between 0 and 20. And I'm replacing letters by numbers here. There are 20 amino acids, so you would expect 20 states. I'm including also a 0, because we have the alignment gap, which is an extra character. You see it here. What is the gap? So it's because when we align sequences that are coming from different organisms, for instance, you may have mutations that have simply substituted an amino acid by another, but you may also have some other mutations that have taken away one amino acid. These are called deletions, or inserted an amino acid. These are called insertions. Because of deletions and insertions, it's necessary to include gaps in the alignment. So the way to best align protein sequences is a whole field. I'm not going to spend time on this. I'm just telling you we assume that these sequences have been aligned, and because of this, all the amino acids in a given column have the same position, for instance, on the 3D structure, essentially. And so that's one of my columns, and you can have the alignment gap because of this alignment. Okay, so that's the idea. And the one-body statistics, I'm going to focus on this column I, and to study the frequency that I observe the amino acid alpha I at this column I. And this frequency is just going to be the number of sequences in my alignment in which there is this amino acid divided by m, which is my total number of sequences. So for instance, here, in the column I highlighted, you have frequency 1 of having H, frequency 0 of having all the other ones. You have other columns which are less, which have a distribution which is more complicated. For instance, this one, you see a lot of T's, but there is an N. So okay, N would have a smaller frequency than T, stuff like this. So that's one-body statistics. I would like also to emphasize that if m goes to infinity, which of course we can never achieve in real data, but if m was to go to infinity, this would tend to a probability distribution of the one-body statistics, which I will call pi of alpha I. Okay, and next, we can, in fact, represent this information about the one-body statistics in what is called a sequence logo. So you can see here the logo. So let's first observe it. So you can see here this corresponds to the column that I highlighted. You can see a very large H. And in fact, the height of the letter here is proportional to the amount of, to how often you observe this H, and more specifically, it's something that is defined from the entropy. So let me introduce entropy. So we already had, in fact, discussions about information theory in yesterday's talks by Hervé Isambert. But I'm going to just do a fast reminder of this. And so, so I'm going to introduce the Shannon entropy. And for this, I'm going to look at a random variable, because it's, so let's consider a random variable, because it enables me to introduce the formal notion, and then we will apply it to a protein, to a column of the alignment, a random variable x. So the entropy, which I will note h, of this random variable is equal to minus the sum over all possible values that x can take. And I will write x in omega, so this is the set of possible x. So x taking values x in omega of p of x log 2 of p of x. Okay, and so this is something that, it's a quantity that has the advantage of depending only on the distribution, p of x, and not on the values, for instance, that x can take. And this is very useful for categorical data, such as this protein sequence data, where you have a set of amino acids that you can map to numbers. And here I did it, like from 0 to 20, but the mapping is arbitrary. Like there is no reason why alanine would have number 0 or whatever, So it's nice to work with quantities that don't depend on this mapping, and this one depends only on the probability distribution. And we can approximate it by looking at the frequencies. There are some important finite size effects which I will not discuss in this talk. Yes, I see that there is a question.
Yes, x-axis and y-axis, what are they?
Sorry, okay, I didn't hear the full.
Yeah,
sorry, okay. So I haven't fully defined this yet. I'm just introducing the way we define the height for now. The x-axis is just the number of the columns. So we start with column 0. Column 0 is this one. Then column 1, column 2, okay? So that's why, I've written this line which associates this column to this one. This is number 10, starting from 0, and this is the 11th column. And then the height is related to entropy, which is what I'm explaining now. You're welcome. Okay, so that's the entropy of a random variable. And, we can take the simple example, so, of the uniform distribution. So if the number of possible values, cardinal of omega, is n, and if p of x is equal to 1 over n for all x in omega, so that's a uniform distribution, then h of x is going to be, well, minus. So we have n times the same value, so n times 1 over n times log 2 of 1 over n. And this is equal to log 2 of n. So that's the entropy of a uniform distribution, and you may notice the similarity with the Boltzmann entropy that you're used to in statistical physics, which is Boltzmann constant times natural logarithm of the number of states. Okay, so this is the same, except that we have a multiplicative factor coming from this use of the base 2, which is common in information theory. Okay, so that's, in fact, the maximum value that entropy can take. And to illustrate this, I will take the very simple example of the coin. And I will take a biased coin. So the probability of getting heads, which I will denote by 0, is called P. And the probability of getting tails, which I will denote by 1, is 1 minus P. In this case, the entropy of this random variable, which is the outcome of the coin toss, is just minus P log 2 of P minus 1 minus P log 2 of 1 minus P. And if you plot this, the entropy of X versus the parameter P, you will find that, so P is between 0 and 1, you will find that this entropy has a value that is, okay, I mean, my curve is badly drawn, sorry. All right, I will do this, okay, right, this way. All right, so it's something like this, and the maximum of this entropy is obtained for probability equal to 1/2, which is the uniform distribution case, same probability of having heads or tails. And the minimum is obtained for P equal to 0 or 1, which is the case where you are certain that your coin is going to fall either on heads or on tails. It's a distribution where there is no uncertainty before you toss the coin, you already know the outcome. So this gives us, in fact, an insight into the interpretation of entropy. Entropy is the information that you are missing about the outcome if you know the probability distribution, but you don't know the outcome yet. And so, for instance, if you consider now a variable which is coming from an experimental measurement, which can be a measurement of anything, and you measure a histogram, and for instance, there are 4 possible values. Let's take this, like 1, 2, 3, 4. If your distribution is something like this, almost flat, so this is almost 1/4. If you observe this histogram, so that is x, that is P of x, then your entropy is almost maximal. And if you have a histogram that is close to, I mean, that is very peaked, something like this, then your entropy is very small. Sorry, very large. Sorry, very small. Okay, sorry, I'm confused. This is, so if it was just one big peak, we would have an entropy 0 as here. And here it's like it will be small, and this one will be close to the maximum, which here would be 2 bits. In the case of the coin, it was, if it's fair, it was 1 bit. So this is, I should write h of x is in bits. And so, okay, so this was 1 bit, and in fact, for a coin, you can understand this by noticing that, in fact, to get the outcome, you need to ask only one binary question, yes or no. This is what it means to have 1 bit of information. Okay. So that is entropy, and the height of our logo is going to be, in fact, the maximum possible value of entropy minus the value of entropy in this column. So if we consider a column, so in a column of our alignment, we have the entropy in column I, let's say. Column I. The entropy Hi is going to be minus the sum over alpha I of Pi of alpha I log 2 of Pi of alpha I. And we are going to approximate it by the same thing with the frequency that is measured on the data directly, log 2 of Fi of alpha I. And that is only true if m is very large, so in the limit m goes to infinity. And that is entropy again, and the height of the logo is the maximum possible value of entropy minus the actual value of entropy. And so here, the maximum possible value of entropy is log 2 of 21, because there are 21 states, minus hi. And so that's the height that is plotted here. Okay, so you see it ranges till 4 point something. And at this site number 10, we have a height that is equal to the maximum, because there was only one possible outcome. So we had hi, which was equal to 0, and the height, which was equal to log 221. So that's the height. And you can see here that this column has a smaller height, which means that the distribution has more entropy. And in addition, the letters that are shown here are proportional to the frequency at which each letter is observed. For instance, here it means that S is the most common amino acid in this column, A is the second one, etc. So that gives us really a lot of information about the one-body statistics. Okay. But then you may ask, how good is it if we know only the one-body statistics? And we are going to see that, in fact, it's not sufficient to specify a protein fold. And for this, I'm going to show you results from a very nice experiment from this paper here, which looked at a protein domain and made some synthetic example of this domain. So they started by looking at some natural instances of these domains in different species and expressing it in a common system. I will not go into the experimental details. Here you can see that 67% of these natural sequences were folding properly. Okay? So you may be a bit surprised that it's not 100%. This is due to the experiment that expresses them in another organism, plus this is a domain, which is a piece of a protein, so it's not completely obvious that it should fold properly, but most of them do. All right. Now, in fact, this, I told you already about it. If you consider now a set of random sequences of the same length, none of them folds, right? So folding is something special, a characteristic of natural proteins. But now let's look at what happens when we keep these one-body statistics that I told you about. So the logo is kept. What happens is that still none of these proteins folds. It's a little bit better than before. More of them are soluble, but they don't fold. Okay. And now, in this paper, what they also did was preserving the two-body statistics. Oh, sorry. Preserving the two-body statistics. And here you can see, this time we have some red. 28% of these proteins, which are synthetic sequences, but preserving the two-body statistics, actually fold. So the two-body statistics are really very important for defining a protein fold, and they can be sufficient. So it's not as good as the natural ones, but we do have some folding. So because of this, I will say a few words about two-body statistics. So this time, statistics. All right. So this time we are going to consider two columns in my alignment. So again, this time we take A, but we also take another one, which I will call J. Okay, so this is L. All right. And we are going to measure how many times in the alignment we observe amino acids alpha I and alpha J at the columns I and J. Right. So that's the analog of this quantity, but for two-body, for instance, well, okay. How often do I observe alanine here and histidine here? That's my count. Okay. So, and again, for m goes to infinity, this would converge to a probability distribution, Pij of alpha I, alpha j, which is a theoretical construct because we cannot really make m go to infinity with real data. If we were to consider now two random variables, so here I started with one random variable which corresponded in the end to the state of my column. Now I'm going to take two random variables in order to illustrate what we can do with these two-body statistics. So consider two random variables, x and y. So we can construct, for instance, the covariance between these two random variables. And we can construct it by saying that it's this quantity, average of the product minus the product of the average. But here, you notice that there is something that we will have trouble with for our proteins, is that this quantity depends on the actual values of x and y. And for the protein, again, amino acids have different properties, and it's not obvious that we can make a mapping to numbers, right? We have this problem of the categorical data. We are going to consider, something that is considered in the field is this kind of quantity, which is inspired by this one, but which is defined for each pair of amino acids, alpha and alpha j. And it's just you take the frequency that is observed, Fij of alpha I, alpha j, and you subtract Fi of alpha I times Fj of alpha j. So it's something that is kind of similar to this one. However, we have 21 times 21 values for each pair ij, right? So it's a larger matrix. But this is important nevertheless. And I showed you before that using information theory, we can have a quantity that depends only on probability distributions and not on the values. So here, in fact, for two-body statistics, we can do something similar. And this was, again, something that Hervé introduced in his talk here. It's mutual information. And so let me... Mutual information. So again, so we have these two random variables, x and y, and mutual information is denoted by I. This is the sum over all the possible values that x can take and all the possible values that y can take. So I should write x in omega, y in omega prime. I will just go a bit faster. Times the probability of observing x and y, so a joint probability, times log 2 of p of xy divided by p of x, p of y. So that is mutual information. You can also express it as the Kullback-Leibel divergence between these two distributions, p of xy and p of x times p of y, which gives you a kind of sense of how different these two distributions are. In particular, if they are equal, if p of xy, so if p of xy is equal to p of x, p of y for all xy, then, well, what is in this log is 1, so this is 0. Mutual information is 0. So mutual information is 0 for statistically independent variables. And in fact, mutual information is something that quantifies the amount of information that is shared between the two. And we can see this better by writing this. Okay, so for this I will not do the proof, but it's not a hard proof. We can write, in fact, the entropy, for instance, of the variable x as a decomposition, which is the mutual information between x and y, plus the entropy of x knowing y. And this is the information that is missing about x before you make the draw. This is something I told you about before. And this is the information that is still missing about x if you know the value of y. Okay? And, okay, so I will write this. So, information still missing about x if y is known, and it's on average, right? It's average over all the possible values on average. All right. And so I of x and y is what is learned about x by knowing why. By learning, well, okay, when learning, let's say, when learning. And in fact, it's symmetric. It's also what is learned about y when learning x. So that's something that really quantifies this statistical dependence between two random variables. And I also, I already told you that, well, covariance is something that is probably more usual and also informs you about statistical dependence. However, mutual information has this advantage of depending only on probability distributions, not on the values of x and y. That's already something that is very good. But there is more. Okay, so this is just a Venn diagram showing the relationship to conditional entropies, etc. I will not go into the details, all the more that Hervé presented this yesterday and also made the extension to multivariate cases. All right, so I will just show you this example, which shows three sets of pairs of random variables x and y, for which each dot corresponds to a draw of x and y. And you can see here that there are some statistical dependencies between these variables. What I mean by this is, for instance, if I tell you, okay, I don't know what y is yet, but I'm telling you that x is close to 1, well, you already know that y is probably also close to 1. There is some noise, but it's giving you some information, right? Here too, if I tell you that x is close to 1, you know something about y. So from the point of view of intuition, there is some statistical dependence in all these three cases. However, if we look now at the values of covariance or correlation, well, there is a bit of a problem here. I mean, it's not a problem, it's a fact, but okay. We can see that correlation is very high for this one. Okay, it's a linear dependence, so it's kind of natural. It's close to this maximum value of 1 because correlation is normalized covariance. However, correlation for cases 2 and 3 is extremely close to 0. And that means that correlation is not able to capture this kind of dependencies, which are nonlinear dependencies. At least it's not able to capture all of them. It's focused on the linear cases. And mutual information has the huge advantage that it also captures these types of dependencies. You can see here that the mutual information in these two cases is substantially above 0. Okay, so mutual information is a nice quantity to have a better understanding of the general statistical dependence between two random variables. And it's something that can be used for proteins. And we are going to see later in the talk that it's something that is important. So now I have introduced the notions that I will use, and I can move on to applications. And so you can see here that I have three applications. One of them is how we can use this kind of information to learn about structure. The second one is how we can use it to learn about interactions between proteins. And finally, we can also look at collective modes of correlation. So let's start with structure. And structure is something that we've heard of also in previous talks, especially with the recent advances of AlphaFolds. But here I will tell you about some methods that were introduced before that and that used the kind of information that I'm showing here, like especially the two-body statistics. And so the idea is that if a protein folds into a specific 3D structure, these two amino acids, for instance, here are in contact. So they need to be complementary in terms of their physics and chemistry for this to happen. And if during the course of evolution a mutation changes the chemical nature of one of these amino acids, which is represented by a change of color here, then the interaction is impaired and the structure of the protein is also impaired, which is bad for the fitness of the organism. You can see here the fitness in my simple schematic is decreasing. And here, if you have a complementary mutation that happens here, which is okay, not very likely, but if it happens, we will have a restoration of the complementarity, and then the fitness will go back to a higher value, similar to this one. So what it means is that during the course of evolution, natural selection is going to eliminate this state, which is not folding properly and not functional, but to keep these kinds of states, the first and the third one. So now, if we observe a set of sequences which have this structure and which have evolved from common ancestor, etc., we will observe, in my simple schematic, either red-red or green-green at these two sites. So that's a strong correlation between the states of these two columns. Okay, and you can think, okay, that's great, then we can learn about structure directly by looking at correlations. And so in fact, it's not true, like there are some complications, in particular the fact that correlations can be indirect, so you have to disentangle direct from indirect correlations, and also the fact that not all correlations come from actual functional constraints. Some of them can come just from the evolutionary history. For instance, if you had 2 mutations that happened early at the stage of the ancestor, then this will yield a substantial correlation without any functional constraint. Okay, so things are a bit more complicated than they sound, but this is the basic idea. And a method was developed in order to improve over correlation and also over mutual information, in fact. And this method is based on constructing a maximum entropy distribution for a protein family. And it was first applied to proteins in this paper. But it's a method that is also very useful for other fields, for instance, neuroscience and also others. Okay, so I think before, well, okay, I will say a few words on this before moving to the board. The idea is that we want to find a probability distribution of observing a given sequence within a given family, that is to say, a sequence that will fold in the proper structure and will have the proper function. And this probability distribution is in principle an L-body probability distribution, because, okay, here L is again the length of my protein. And we are going to take a data-driven approach for this problem and to construct this distribution from the data. And for this, we are going to measure in the data the statistics I told you about, which are one-body frequencies and two-body frequencies. And from this, we can get the covariances, for instance. I mean, that is the generalized, I mean, these ones, the CIJs. And we are going to stop there, which means that we are not going to take into account the three-body or higher order things. And you may wonder why. So first of all, we saw in this experiment that I showed you that the two-body statistics can suffice to specify the fold. And the second motivation is that it's hard to measure with a good precision the three-body or higher correlations from from the number of sequences that there are in a given alignment. So this is getting less true now, but okay, it's something that, let's say, it's less precise, okay? So the advantage of the one and two-body frequencies is that there are fewer of them to measure. Already, I mean, the two-body frequencies, you have 21 times 21, which is a big number, right? But three-body would be worse. All right. And so we want to find a probability distribution, an L-body probability distribution that is consistent with the observed statistics at the one and two-body levels. And this means that we want this distribution to marginalize to these distributions if we marginalize over either L-1 for the first one, this one, or L-2 variables. We want to be constant with this. All right. And so the problem is that there are multiple distributions which are consistent with these choices. So how do we choose one? One possible solution is to use the maximum entropy principle. And the idea is that it yields the least structured model that is consistent with the observation, so essentially the most random possible, apart from these constraints. And so for this, we maximize the quantity that I introduced here, which is this Shannon entropy, but under the constraints that are given by these one and two body frequencies. And so I'm going to show you this in the case of one simple example, which is again working with one random variable. Because I think it's, it helps to understand the form that the distribution takes. So that's a very short maximum entropy introduction. And so the idea is that you have partial information about a random variable. For instance, you've measured its average, or maybe the average of its square, something like this. or maybe you've measured absolutely nothing, okay? And you want to find the distribution that has the maximum randomness given consistency with these things that you've measured. And so you want a distribution as random as possible given the constraints. So you maximize entropy under these constraints. So you take the Shannon entropy, h of x, which is this, as I introduced before. All right? And we are going to maximize it under the constraints. So first, let's imagine that we know nothing about our random variable. All right? So 1, we know nothing, which means no measurement has been taken. So the only constraints that we have is that probabilities should be normalized. So the sum over x of p of x is equal to 1. That is our constraint. And we are going to maximize this entropy to find the distribution p of x that maximizes this entropy under this constraint. So for this, we introduce h tilde, which is the entropy with the constraint of this introduced by a Lagrange multiplier. So in order to have a nice thing, I will write lambda divided by log 2, but that's just formal. We can do it without it. Sum over x of p of x. Okay, so we have this variant of the entropy that includes the constraint. The idea is that if we were to maximize with respect to lambda, we will obtain back our constraint that the sum of p has to be normalized to 1. And so now we can maximize this with respect to p of x. And so, okay, I think here I will, just for the sake of clarity, I will say take, A equals p of x. So that's a specific, value of the probability of obtaining a specific x, okay? And we will differentiate the entropy with the constraint h tilde with respect to A. And if you do this, well, okay, so you have to differentiate this sum, but there is only one term of the sum that includes the right x, okay? And so you focus on this one, the other ones will be constant. And you obtain minus log 2 of A, which is your p of x. And you also obtain something from the other term of this product, which is 1, and except that it's a log 2, so it's 1 over log 2. And here, I have then the derivative coming from the constraint. Again, in this sum here, there is only one p of x that is equal to A, because it's a specific x. And you have, because of this, minus lambda divided by log 2. So log 2 of A is, in fact, natural logarithm of A divided by natural logarithm of 2. And so we can say that if it's 0, so dh divided by dA equal to 0 means that, it means that A is equal to the exponential of minus 1 minus lambda. And now, so A is equal to p of x for a specific value of x. That's how we introduce it, okay? So this means that, in fact, for all x, this is true. So for all x in omega, the possible values, we have p of x equal to exponential of minus 1 minus lambda, where lambda is our Lagrange multiplier. And if now we re-implement the constraint that the sum of p's have to be 1, we find that if the cardinal of omega is n, then this is 1 over n, right? So this gives you the value of lambda. I mean, you can find your Lagrange multiplier with this, but it's not really something that we will focus on. We just find a uniform distribution. And I told you before, the uniform distribution is the one with maximum entropy, and we showed that in the case of this coin, it was true. And you can see here that if we know nothing about our random variable, we measure nothing, indeed, the maximum entropy distribution is the uniform distribution. So that's consistent with what we saw before, and that generalizes it. But now let's imagine that we have measured the average value of x. So we have measured average value of x, and that we have found that it's equal to a value m. So now we need to add an extra constraint. And our h tilde is going to be the same thing as before, still the constraint of normalization. So this one. But now an extra constraint, which I will implement with the Lagrange multiplier mu. So it's m minus sum of x p of x. Okay, sorry, it's a bit small probably. All right. And now, if we differentiate this and we do exactly the same process, I will not do the full calculation, but we obtain the priority p of x. So by maximizing this quantity, we obtain p of x equal to exponential of -1 minus lambda minus mu x. Okay, so that's the distribution that you would get. Now, if you measure also the average of x squared, you would obtain here a new x squared. So the idea is that you get in the exponential the terms that you have constrained. And this is just, a simple example that I wanted to show you to show the construction, the way these distributions are constructed. But now we are going to turn back to the case of proteins. And in the case of proteins, if you do the same process, but this time with your L-body priority distributions, then you get a maximum entropy model consistent with these observations, which is given by this form. And you can see the exponential form, which is very similar to my toy examples, with these one-body terms coming from the fact that you have constrained the one-body frequencies, and these two-body terms, which come from the fact that you have constrained the two-body frequencies also. And this distribution is something that physicists recognize as being the Boltzmann distribution associated to a Potts model with these fields and these direct couplings. And this is what was used in this paper, and it's called direct coupling analysis in the field, because it focuses on these couplings. All right, and so, okay, this is just like a bit more on like the fact that we have the constraints here that we marginalize over all variables except one, or all except two, and that's how we get this distribution with the Lagrange multipliers, etc. But I would like to emphasize that getting the actual values of h, I, and eij that match these constraints is in fact a very hard problem. And many approximation methods have been used in order to solve this. I will not go into the details. I will just tell you about the crudest approximation, which is the mean field or small coupling approximation, where the eij's are just obtained by elements of the inverse matrix of covariance. So the tij's are exactly these ones. Okay? So you have a 21 by 21 matrix for each of them. In fact, one is redundant, so it's 20 by 20, but let's not focus on this. Essentially, you inverse this matrix, and you obtain an estimate of the couplings eij. And it turns out that in the field of protein, focusing on these couplings has been a big advance. In fact, the amino acids columns ij that tend to have large values of these couplings are those that tend to be in contact. And so let me show you this. So having a large value of eij is a better predictor of 3D contact than having a large value of cij or of mutual information. So here, okay, maybe we can start by this plot. So this plot, well, okay, no, let's start with the example. So let's consider this example, which is coming from this paper. So here you have the predicted contacts in red or green, which are coming from taking the top pairs in terms of the values of this. So those that have the high eij's. So of course, at this point, you may ask, okay, but the high eij's, did it for a given alpha or beta, etc. So there are different ways to reduce this score to just one score for a pair of column ij. One of them is just to take the norm of this whole matrix. So let's imagine that we do this. We get a score for a pair of columns ij, which is the norm of all these small eij's of alpha beta for all alpha and beta. Okay? So if you take those sites that have the top values of these couplings, and you plot them on the 3D structure, which is here coming from an experiment, that's just to check the predictions, right? You find that most of them are below a certain threshold of distance, which is in red, and only one, in this case, is above the threshold of distance, it's in green. So this one is a wrong, this green one is a wrong prediction, and all the red ones are correct predictions. So that's a pretty good amount of correct predictions. And it is better than focusing on the top with, on the set of top pairs with top mutual information. So it means that this maximum entropy approach is giving you better results. And this is confirmed on this plot, which is averaged over multiple protein families, where you get the mean fraction of correct predictions versus the number of predictions that you make. You can see here the results from the maximum entropy method in black and the results from mutual information in red. So you see that the black curve is getting better than the red curve and also better than the yellow curve, which was another method based on a Bayesian dependency tree. Okay, so that's a good success of this maximum entropy method. And it has been used in order to make full predictions of 3D structures from sequence data. So you need for this an alignment, you build the model, and you use the constraints given by the contacts to build your structure. You can see here the predicted structures versus the actual structures from X-ray crystallography, the crystal structures. And you can see that in these three cases, the predictions are really good. Okay, like what I mean is that this and this are very similar. In each case, you have two views of the same proteins, which is why you have these two copies, just to see it better. I mean, I'm mentioning this in case this is not clear. I hope it's fine. Okay, so good predictions of structures. However, there are some limitations, and the limitations include the fact that it's necessary to have a large number, M, in fact, of homologous protein sequences for this to work well, simply because we need to learn well the covariances, theCij's of alpha I, alpha j. And for this, we need substantial data. We also need this data to be sufficiently diverse. Imagine a protein that would be so conserved that it would be exactly the same in all organisms, then we could not learn anything about covariances, right? So that would be also bad. So we need a lot of data and a lot of diversity. And this means that despite the fact that this method is very nice, it cannot be applied to small protein families, which are restricted to, for instance, a small subset of living organisms, or stuff like this. This method, nevertheless, has had other great applications than structure predictions. For instance, recently it was used in order to perform actual protein design in this paper. It's also something that can be used to infer which proteins interact together, and I will mention this a bit later in my talk. It can be used also to predict the effect of mutations and to describe protein evolution. However, because this is still my section on structure, I would like to mention that now there have been some great advances brought by deep learning in the field. And this is something that you heard about already in the lectures by Christophe Zimmer and Giuliano Ziconacci yesterday. But I will just say a few words on this. So these approaches won the contest of protein structure predictions, the two last ones, and especially the last one in 2020 by a spectacular margin. They employ methods that are very much based on natural language processing types of approaches, which is that they use the attention mechanism and transformers. I will not go into details. And I want to emphasize that this is a supervised model, meaning that it has learned from known experimental structures given as input, and also that it is one model, a very big model with a lot of parameters that is built for all families and all proteins, while the maximum entropy one is building a specific model for a given protein family. So really different kinds of models. And here I'm showing you the performance curve that you saw in previous lectures, so I will not go into the details. Performance is very big, okay, fine. And I want to also mention another model, which is interesting because it's similar in terms of employing natural language processing techniques, but it's unsupervised in the sense that it doesn't learn from structures. And nevertheless, this method has a good performance, and here it is compared to the POTS model, which is the maximum entropy model that I introduced before. And there are two variants. One of them in orange, which does not include alignments or coevolution, and one in blue, which is performing better because this is like the fraction of, the percentage of correct predictions, that includes the multiple sequence alignments, and so it has information about coevolution. And you can see that these models outperform the POTS model, especially for small alignments, and I told you that small alignments are the ones that are problematic for maximum entropy. So it's kind of interesting to see these advances, and Damiano in his poster mentioned more about this. All right.
So what does it use?
Sorry, what is?
What does it use, this transformer method, if it doesn't use structures?
So it's okay. So the idea is that it uses the masked language objective. So like the idea is that you have a sentence and you mask a word and you want to be able to infer this word. So the objective of training of this network is this, and it turns out that it's able, nevertheless. I'm not sure if it's nevertheless, but it's able to learn structures.
But it learns on sequences?
Yes, it learns on sequences. Sorry, yeah. The training is sequences. So in this case, sorry, I lost my pointer. Especially in this case, the MSA transformer, it takes multiple sequence alignments as input. So it learns from sequences, yeah, indeed.
And then what model does it, then I guess it's the same as a POTS model to go from, to actually predict the structure. What does it do? It needs to do some molecular dynamics or something.
The idea is that, okay, it's able to capture the signal from the coevolution and to get the structure, but it's not explicitly using the POTS model. It's just, taking the multiple sequence alignment as an input.
Right, but then you're doing structure prediction, so how do you generate structure from?
Okay. So the structure is found from looking at the attention heads in the model, and in order to improve it, it's possible to make like a very slight training, like to training on a very small data set to improve the combination of attention heads that you will use in order to make the prediction. So in that case, you could consider it's not fully unsupervised, but the supervision is really very small compared to AlphaFold, which took all the. So very good point. In order to make it the best, you kind of need to find out which attention set to use. Yeah. Any other question? All good? Yes. Question.
On AlphaFold, if you could show the slide on the previous slide.
Sorry, I didn't get it.
Yeah, the previous slide.
The AlphaFold, sorry.
Yeah.
This one?
Yeah, this one, yeah. So if you look at the AlphaFold 2,
yes.
So obviously it's very impressive, almost 90%. This is sort of average over all proteins they did.
Look, I think in this particular plot, I'm showing the score in the free modeling category, which is like the ones for which, the targets that are considered to be the hardest, the ones for which there is no, like the homologs are super distant, and there is, it's considered that there is no knowledge about structure even from.
So sorry, there is one protein here on this.
Sorry, there is one.
There is just one protein on this.
It's sorry. I don't remember the number, but it's the average on the free modeling category.
And you
so there are a certain number of targets in the CASP contest, and they are subdivided into difficulty classes. Yeah.
Now I was wondering what is the error.
Yes. No, that's a good question. Yeah.
I don't know.
I cannot. Yeah, I don't know either. But I think there is some variability. Yeah. But, yeah. Sure. Sorry. Yeah. I don't have the plot for this. All right. Okay. So I will now move on to another application of these statistics and what we can learn from protein sequence data about function of the proteins. And this time it's not going to be about structure, but about interactions. And this time it's work that I have participated in. And the idea is that we want to find out which proteins interact together. And this is a very important problem in biology because protein-protein interactions are crucial for the stability of multi-protein complexes, which carry out important functions in the cell. And these interactions are also crucial to maintain the specificity of signaling pathways. However, determining them experimentally is quite complicated, especially if you want to do this for a full network. There are many proteins in an organism, so it's a hard task. And also there are many species. Even if you do it in one species, it's not obvious that everything will transfer to another. And even in E. coli, which is the case looked at by this paper, the full protein-protein interaction network is not fully known experimentally. There are still some, uncertainties. All right. So it is a good motivation to try to learn about these interactions from protein sequence data. And here I'm mentioning like some examples of papers that have focused on this. I will not go into the detail, and this is not exhaustive, of course, it's just some examples. So what I want to say about this is that many approaches can be used in order to find out about interactions from sequences. You can focus on phylogeny, on sequence coevolution in the spirit of what I told you before. And this is what I will tell you about more, but I just want to mention that it's not the only possibility. So phylogeny is also possible. You can also just assemble all types of evidence, including co-mentioned in the literature, etc. This is actually something that works quite well. And you can also use machine learning approaches. And more, right? But just wanted to mention that the problem can be addressed in different ways. But now I'm going to focus on the ways which use coevolution. And here you see a schematic which is quite similar to what I showed you before in the case of folding, except that now you have two protein chains, A and B, that interact together. And the points of interactions are going to be correlated to one another, right? Because again, if you have a mutation that happens on one of these amino acids, it's going to impair this time the binding between these two proteins, which is important for their function. And this is going to be selected out by natural selection. However, if you have a complementary mutation, it can restore the complementarity. So if you have two alignments of these protein families A and B, where the interacting partners are known and they are in front of one another, if you have this, then you will observe correlations. However, knowing which A interacts with which B is not something that is easy in general. If you have only, let's say, one copy of A and one copy of B per genome, and you know that they interact in already a lot of them, then it's fair to assume that you will have an interaction that is more general. But if you have several copies of A and B in each genome, called paralogs, then knowing which A interacts with which B is difficult because all the A's are kind of similar, they are homologs. All the B's are also similar, so those A1 interact with B1 or with B2. That is the question I'm going to address. I just want to mention that there is another question that can be addressed with similar methods, which is, if I give you two protein families, A and B, do they interact together or not? Imagine we know nothing about it. So that's another question that is very important. Part of it can be addressed with this kind of method. But I will focus on this one because it's easier to explain. And for this, we proposed an iterative pairing algorithm, which is based on direct coupling analysis or maximum entropy methods. And we want to address this question, does A1 interact with B1 or with B2, etc.? And we may or may not have a training set of known interacting partners. But let's imagine that we do, right? If we do, we are going to build an alignment of these sequences where we put the sequence of A and of its partner B as a long sequence, okay? So it's like A and B, concatenated sequence. And below, we put, again, another pair that is known, A and B. And from these data, we can learn, again, the Cij, these generalized covariances or correlations. And we can construct a maximum entropy model, which will give us direct couplings, but this time for these long sequences. And we use this in order to define a score, which is the effective interaction energy for each possible pair, A1B1, A1B1, A1B2, etc. And this score is used in order to make predictions. We also take into account then the best ranked predictions by our model, and we include them in our concatenated alignment, which is then used to build a new model which has more information into it, and which hopefully is going to be better. I say hopefully because if we include a false, like an incorrect prediction, then it's not going to get better. But okay, we hope that the best ranked predictions are correct and that the model is going to improve. And we iterate over this process. And the idea is that this iterative algorithm approximately minimizes these effective interaction energies between partners. And I just want to tell you what is exactly this effective interaction energy. So for this, I mean, here you can see this maximum entropy, pairwise maximum entropy model that I introduced before, the EIJs, which we take again in this crude approximation to be the inverse elements of the covariance matrix. And we can construct this score, which is the sum of the pairwise interactions between sites I and J, sum of the EIJs, where I is in protein A and and J is in protein B. So we really focus on these interaction terms. And we use this score in order to rank our possible pairs, A1, B1, A1, B2, etc. And so that's the score that is used in order to make the pair assignments and to rank the pairs. Okay, so now let me show you how this, okay, I think I will not, well, how this method performs on a benchmark data set. So just 2 words on this benchmark data set. It's a set of real protein sequences from prokaryotes, so essentially bacteria, which includes 2 protein families, kinases and response regulators, histidine kinases and response regulators. So the histidine kinases are proteins that to which a ligand can bind, and then they autophosphorylate and transfer a phosphate group to their cognate response regulator. And this triggers a response by the cell to the initial stimulus. So it's a signaling pathway with just 2 components. Okay? And these 2 proteins have a high specificity of interaction, especially given that each bacteria has several such systems which are aiming to respond to different stimuli. And so here we are going to address the task of finding from sequences which ones interact with which ones. And it's a task for which we know the answer from genome proximity, so it's a good benchmark. And here I'm showing you the performance of the method in terms of the fraction of correctly predicted pairs if we make a number of prediction which is equal to the total number of pairs. I mean, we also looked at ROC curve, but okay, this is one score. And it's plotted versus the effective number of pairs in the concatenated alignment, which grows as we iterate here. So as we iterate, we add more and more sequences into this alignment. And so it means that here I'm plotting versus essentially advancement in the algorithm. So as we progress, as we iterate, we see that the fraction of correct predictions is increasing, which is good, right? And here we also have results that are better than the chance expectation, which is the one that is obtained by matching randomly A's and B's. And you can see here that the results also depend a lot on the size of the training set, so the size of pairs that we know at the beginning, okay, those that are given. Having a large training set is better, which you may have expected. And here, if we have a very large training set, so 2,000 out of 5,000, we see that the performance essentially doesn't improve much when we iterate, while if we have a small training set, it improves a lot. And this is summarized by this plot on the right-hand side, where the fraction of correct prediction is plotted versus the size of the training set, both without iterating in blue, and at the end of the iterative procedure in red. So you can see that without iterating, we need to have a lot of data in the training set to get a good model and to make good predictions. Okay, we need a lot of data to learn the CIJs, etc. But thanks to the iterative procedure, it's possible to make good predictions even in the case of small training sets. And in fact, even in the case of very small training sets, down to 1, and we, because of these results, we found a method, in fact, to make predictions without a training set. I will not go into the details, but it's possible to do this. And now, I would like to mention that for this part, I used the maximum entropy model, the EIJs, in order to build these scores. But then, something that I found out later is that it's possible to also use scores based on mutual information for this. You may remember that for the prediction of structures, the maximum entropy model was really outperforming mutual information. But it turns out that for this task of pairing, interacting proteins, mutual information does quite well. And so this is a variant of the iterative pairing algorithm, which uses a score based on mutual information instead of the maximum entropy model. And here, you have the comparison between the two models. So one is in red and the other in blue. So mutual information is in red. You can see that mutual information, after iterating, performs slightly better than maximum entropy, but just slightly. It's very similar. Mutual information is also a little bit less data thirsty. You can see that at a given amount of data, performs slightly better. And this is something that is quite interesting. I mean, the fact that mutual information performs badly for contact prediction, but well here, is interesting. I mean, it's coming from different ingredients. One of them is that for structure prediction, we need to disentangle direct from indirect correlations. Here, it's something that doesn't matter so much. We just want a score, right? A global score, I mean. And also, mutual information is something that is agnostic about the origin of these statistical dependence. It just captures all statistical dependence. And it also doesn't require an inversion of a matrix, which means that it's a bit more robust to small data sizes. So different reasons. But so yeah, mutual information performs quite well here. And we also found potential signatures of the existence of an interaction between two protein families A and B using this kind of methods. So that's about it for protein interaction predictions. And I see that, okay, I still have maybe a bit of time. Yes?
I mean, I suggest 10 minutes of talking and then maybe 5 minutes.
Okay, 5 minutes. Thanks. I will do a fast tour of this.
You can talk for 10 minutes and then 5 minutes for questions.
Okay, I will try to not go over too much. Thanks. Yeah, so another thing that we can look at from these multiple sequence alignments is collective modes of correlation. So far we focused on, for structural two sides. These are one and two body statistics, but we can go beyond this. There are multiple ways to do this. So one of them would be multivariate information. And it's something that, we are interested in, but I will not talk about in this talk. Another way is to focus on this matrix, the Cij matrix, and to make a decomposition in eigenvalues and eigenvectors to look at collective modes. And this is something that has been studied, and in this paper, the notion of sectors was introduced, so starting from a covariance matrix, but reweighted by conservation, so focusing on the conserved correlations. So this is this matrix, okay? So red means high correlation. They did this eigenvalue, eigenvector analysis of this matrix and reordered the sites and focused on the sites that contribute most to these top eigenvectors. And you can see that after this reordering is done, you can see blocks of collectively correlated amino acids that appear. And it's interesting to see that if these sites are plotted on the 3D structure of the protein, then they are contiguous in 3D. Which means that they have some structural information inside, while they were built just from sequences. And also they have some roles associated to function. Okay, I will not give more details, but they also have functional roles. And another group did some work on a model system, an elastic system, where you ask for having a certain target deformation on one side as a response to a given stimulus on the other side, or to optimize the cooperativity of the deformation, and you evolve this elastic network for this. And it turns out that in this case, the evolved optimal allosteric materials have some sectors that are similar to the previous one and that emerge. So it's coming from this mechanical optimization, and it's associated to the fact that there is a path between the two sites, and that there is a soft mode of deformation, for instance, a shear mode that is easy to deform, or a hinge mode, or a twist mode, modes that don't cost much energy. And this is relevant for proteins because, in a protein will have to deform, for instance, to bind to a ligand, and if this is not costly, so it's a good thing, right? Okay, so sectors can come really from functional constraints on the mechanics, in this case. And we proposed a method in order to find out these functional sectors from sequence data. And for this, we started from an abstract model where you have an additive trait. So a trait is a property of the protein that is important. And we are not going to say more, it's just a property, right? And we assume, and this is a strong assumption, that this property is additive in terms of the sites L, so the mutational effect add, okay? And then we are going to perform nonlinear selection on this additive traits, and it was shown that this is something that, nonlinear selection on additive traits in general is something that describes well experimental data, so even though it seems like a major simplification, at least in some cases it's a good one. And so far the trait seems very abstract, but we proposed one example in a toy model of a trait, which is the energy of deformation of a protein in an elastic network model. So it's an elastic network model based on this paper, but where we included sequence dependence in a very simplified way. The spring constant of the elastic model is just affected by mutations by being reduced. And this means that the mechanics depends on the sequence, all right? And within this model, we can show that the energy of deformation of the protein is an additive trait for small deformations and small mutational defects. So that's just an example in this toy model. And then we can do nonlinear selection on this additive trait. And so we did this by assuming that selection favors one given value of the trait, which is called delta E star. So the fitness, we took a simple quadratic function that penalizes any deviation from this optimal value. And here, among the possible values of delta E for random sequences, which is in black, we make a selection window, which is in orange, and we select those sequences. And then, inspired by previous work, we look at the covariance matrix and at the eigenvector decomposition of this covariance matrix, the collective modes. So here you can see the eigenvalues of the matrix of the selected sequences, of the covariance matrix of the selected sequences. And you find that there are some outliers, but what is interesting is that the main outlier is in fact the smallest eigenvalue, not the largest, right? While usually the focus is on the largest eigenvalues. If you look at the associated eigenvector to this smallest eigenvalue, you find that in fact it reflects the vector delta, which gives the mutational effect at each site. So it in fact reveals what selection there was on these proteins. So it really reveals the functional selection that has occurred. So now you may wonder why this eigenvector is so important. And in order to understand this, we have to go back to the fitness. So we have implemented a selection such that the scalar product of the sequence S with the mutational effect vector delta is close to the optimal value delta E star that we selected for the trait. And it means, okay, the scalar product has to be close to a given value. If we represent sequences in space as points, it means that they approximately lie on a plane that is perpendicular to delta, so that their projection on delta is the same. And this means that if you look at this cloud of orange points, they have small variance in the direction delta. So delta is the direction of small variance, which is why it corresponds to a small eigenvalue in the covariance matrix. And this is in fact analogous to a repulsive pattern in a generalized Hopfield model. So the small eigenvalue tells us a lot. However, there is a problem that small eigenvalues can be generated by other sources than this one. For instance, if you have a site that is very highly conserved, it means no variation on this site, and this will yield a low variance direction. We propose a method in order to alleviate this problem, which is focusing on the inverse matrix of covariance, which reminds you of this direct coupling analysis, right? But suppressing the diagonal, because the diagonal contains information about conservation. And with this, we were able to show that the method is more robust to conservation. So I will not go into more details on this. I will just mention one last thing, which is that I told you about functional modes of collective correlation. So far, the idea was you select for a trait and you get these collective correlations that emerge, right? Or maybe you select for a mechanical property and you get this collective correlation that emerge. Now I want to mention that collective correlations can also emerge from phylogeny. And so correlations can emerge from optimization. This is what I've told you about in the rest of the talk. You have these correlated mutations, and so the observed sequence that survive selection have these correlations, and this comes from the fact that the fitness landscape is non-trivial, it has some optima, etc. But covariances can also come from historical contingency and phylogeny, because sequences in a family evolve from the same ancestor. And if you have, for instance, two early mutations at two different sites, so here HS becomes EC, then you will have a covariance between the sequences. And this covariance is something that does not reveal anything about constraints on selection, function, etc., just coming from history, and it would exist even if the fitness landscape was completely flat. And this is something that has been, in fact, studied for a long time. And here I'm showing you the results from this paper, which show that if you perform principal component analysis on a sequence alignment for a given protein family with subfamilies, the subfamilies are, in fact, sorted quite well by this principal component analysis, which means that these high variance modes, collective modes, reveal a lot about phylogeny. And in fact, it's something that was also observed in the sector analysis that I told you about before. There is a more formal study that showed in this paper that the top eigenvalues of the covariance matrix of an alignment that has phylogeny, a lot of phylogeny, follow a power law. And this power law is really coming from the structure of the phylogenetic correlation. And so these correlations from phylogeny, now you may wonder how they impact the functional correlation that I've told you about before. And as far as the prediction of contacts and structure is concerned, they obscure their predictions because, it's like, you have another correlation now that is purely coming from phylogeny. It's bad for you, right? So a lot of work has focused on this. There is actually a usual correction that is employed in the field in order to partially correct for this. But you may wonder, what about the inference of interaction partners, for instance? And it turns out that proteins that interact together tend to share a common phylogeny. And because of this, phylogeny is in fact useful in order to predict interaction partners. And here we made a toy model in this paper of neutral evolution. So here we have a random ancestral chain that evolves by duplications and mutations, but all mutations are accepted. So it's purely neutral evolution on my flat landscape, no functional constraints, and all correlations that exist at the end of this evolution are due to phylogeny. And we ask now, if we take a subset of these sequences as a training set and the other as a testing set, and we build a maximum entropy model on the training set, are we able to predict which halves, so which blue and red halves, have evolved together along the tree? So purely evolutionary partners without any functional constraint. And it turns out that this is something that works quite well. So here you just have the matrix of the effective interaction energies, which are the sum of the EIJs as before. You see a blue line along the diagonal, which corresponds to the pairs that have co-evolved along the tree. And also you see an interesting nested structure of blue blocks, which corresponds to the neighbors on the tree. So I will not go into much more detail, but these methods do capture stuff about phylogeny, and for the inference of interaction partners, it's something that is actually helpful, while it's bad for the inference of structure. And actually, you have heard more about this in Nicolas' short talk earlier in this meeting. And with that, I would just like to summarize a bit and say that covariation in protein sequences, so these two-body statistics here, help us to understand quite a lot, in fact, so about structure, protein-protein interactions, and functional sector, but they also include information about phylogeny, which is quite interesting. And with that, I would like to thank a lot of people. So all the people I've worked with on this protein sequence side of my research. So Ned Wingrin from Princeton, Lucy Colwell from Cambridge, Martin White from Sorbonne, Mohamed Barakan, Philippe Hortel from Sera Cadarache, and a number of students who have either done one project of their PhD or a master project on this topic: Rob Dwyer, Shuwen Wang, Carlos Gandarilla-Perez, Guillaume Marmier, Pierre Marny, and Andonis Gerardos. And I also take the opportunity to thank my other colleagues on the side of population evolution: David Schwab, Claude Loverdeau, Darka Labavitch, Loïc Marek, and Irene Lamberti. and my current group members, some of whom are here, Nicolas Dittler, Richard Servajan, and Damiano Scarbosa. They are among you. You may have met them, so they are in green here. And the other ones, Alia Abaha, Umberto Lupo, and Alix Mouawad. And then I also thank my current funding sources, which are EPFL and the European Research Council. And finally, I thank you very much for your attention. And I'm happy to take questions if there is time. Okay.
I think we can do a few questions.
Hi, thank you for the talk. And I was wondering why in the third part of the works you showed us, you organized, let's say, the couplings between variables with correlation for let the sector structure to emerge, and not, for example, mutual information, or it seems reasonable. I mean, all the thought was based on mutual information, and then in the end, one comes back to correlation. I was wondering why.
Yeah, okay, so you're asking why I'm not using mutual information, for instance?
Yes.
So that's a great question. Thanks for asking it. I mean, in fact, it's possible to find out about sectors using mutual information. It's something that actually I participated in such a work which was led by Casey Huang and his group. And in this paper, we actually relied on mutual information, in fact, normalized by joint entropy, but like, okay. But yes, so to answer your question briefly, yes, it's possible to use mutual information to find out about sectors. However, in the literature, it was analyzed first with covariances.
Okay.
Yeah.
But did the same sector structure emerge? Or?
Yeah, great question. So there are some similarities, but there are also some differences. So it's not, I cannot give a very simple answer to this.